Hello dear students, this is our second lecture that is about the evolutionary pathway from the loci to the present, and then we will see about the difference between eukaryotic cell and the prokaryotic cell. And here the cells spelling is wrong, kindly don't mind. And in the third section we will see about the general characteristics or characters of cellular microorganism in three domain of the life. Let me see about the phylogenetic or the evolutionary pathway of the microorganisms. Um, as uh, as we know that according to the symbiotic theory, we know it the prokaryotes are basically forming the eukaryotes. In the symbiotic theory, we see that mitochondria and chloroplast were in the past time they were living inside the body of or inside the cell of any organism. They were living solely, but after the passage of time, due to some factors, you can as in the theory explains that due to for the protection purpose, maybe a lot of others, they were actually engulfed by the bacteria, and the inside the bacteria they make food, or you can say energy for the bacterial cell. Indirectly the bacterial cell provided the shelter, like we see in the symbiotic relationship. So this is what the symbiotic theory is telling us. There is a scientist, Carl Vizzi. Carl Vizzi actually uh, work upon the phylogenetic relationship of organisms. He basically work on the evolutionary relationship between the microorganisms, through which he describe the microorganism by their biochemical, genetic methods, and many others, and their effect on the microbiology. So he was the person that is basically responsible for that actually gave us this graph that is showing to you. So this graph actually telling us that the ancestral prokaryotes were diverged into two basic terms, that were archaea and the bacteria. But these ancestral prokaryotes also give rise to ancestral eukaryotes, because they include the mitochondria and the chloroplast, and these mitochondria and chloroplasts were converted into eukaryotes. And we know it, the eukaryotes contain mitochondria and chloroplast, and these eukaryotes are divided into four types. They were plants, protozoans, fungi, animals. So indirectly this illustration, this diagram basically denotes the phylogenetic pathway from the LUSA. The word LUSA means the last universal common ancestor to the present organisms. So this is the whole illustration. Next, there are two terms that are the prokaryotic cells and the eukaryotic cells. As we know it, prokaryotic cells are the unicellular organisms that were living solely, and they didn't form the tissues like the eukaryotic cells do, and they having different shape, they are in different colors, different characteristics from the eukaryotic cells, and example include the bacterias. While if I talk about the eukaryotic cells, then these are the cells to which I also made up of, and plants and many organisms, like other organisms like plants, animals, birds, chordates, vertebrates, invertebrates are basically made up of the eukaryotic cells. Now the Vizzy basically provide us the insight for the first time as the coherent view of the evolutionary pathway behind the diversity of living organisms, and he gave us the Satisfactory explanation about the existence and the diversity of the prokaryotic and eukaryotic cells explains that all living organisms or the living forms are fall into three basic domains of lives. What are these three basic domains? These were firstly the bacteria, archaea, and the eukarya. And this division is basically done by the Vizi. And this whole paragraph or the whole picture basically tell us about these lineages, that how these were formed and how these were their exploitation basically occur. Now we see about the general characteristics of cellular microorganism in three domains of the life. The second part of this video. The basically I this table shows that the domains and the prokaryotes and the eukaryotes, their general characteristics. First, the major groups having their cell diameter, the gram positive according to the diameter, gram positive proteobacteria, it's like many others, having nearly 1 micrometer cell diameter. And in case of archaea, the methanococcus bacteria, thermococcus bacteria having diameter of the nearly 1 micrometer. And in eukarya, we see fungi, entomociliates, flagellates, they having diameter 10 micrometer, nearly 10 micrometer. Now you can differentiate these three on the basis of this table. You can see here these three were differentiated. These three domains were actually differentiated according to their characteristics. The chromosome, the chromosomes, if we see about the chromosome, then the bacteria is having single closed circular chromosome and archaea having single closed circular. Similarly, not sorry, the eukarya having multiple linear chromosomes, and introns were in prokarya, the bacteria having very rare introns, archaea also having very rare intron, but eukarya having common introns. So there are messenger RNA, ribosomes, and many other characteristics were there on the basis of which these three domains were actually being explained in this table. Now let me know about the comparison between the eukaryotic cells and the prokaryotic cells. The first characteristic is the nucleus. Let me see about the nucleus of the both cells. The eukaryotic cells having true nucleus. Actually, the present and the absent terms actually denotes that eukaryotic cells having the true nucleus, their nuclear membrane, their the material that the nucleus basically defines are totally present in the eukaryotic cell, but in the prokaryotic cell there are some things that is very important for a nucleus are absent, due to which we called it that the nucleus is absent. Actually, nucleus is not absent. Nuclear membrane and the true membrane that basically covers the nucleus are absent. So we called it that nucleus is absent. True nucleus is actually absent. Next point is the cell membrane. Cell membrane were both having the cell membrane. This is similarity. Next, the cell wall. Cell wall in the eukaryotic cells, um, uh, there are some organisms where the cell wall is present, like the plant. Kingdom Plantae contain uh, having the organisms contain the cell wall, but in the case of animals, there is no type of cell wall is present. And next we see in prokaryotic cell wall is present in the prokaryotic cells, but there is the substance that is the peptidoglycan. You learn about the bacterial cell wall where you can see that the peptidoglycan term. The substance is basically present in the bacteria, actually in the gram-negative bacteria. Now this uh, cell wall is basically present in those type of organisms, those type of bacteria that whose outer membrane material containing the peptidoglycan. So mostly the prokaryotes having cell wall. Now the membrane containing organelles that are actually covered with the membrane were present in the prokaryotes but absent in the eukaryote. After this, there are ribosomes. Ribosomes are eukaryotic cells having larger ribosomes, but the prokaryotic cells having smaller. Flagellas are also present in some cells, but uh, in case of prokaryotes, there are also different types of flagellas are present. Cilias were present in eukaryotic but absent in the prokaryotic cells, and the chromosomes were present in the eukaryotic cells. In case of prokaryotic cells, the chromosomes were in the form of the circular structure that were also known as plasmids. Here you can see the diagram of both prokaryotic cells and the eukaryotic cells. Everything is similar, I think so, and you also observe it. But there are few things are, you can see the basic thing is that their nucleus. You can see in case of animal cell, the nucleus is in the center and it is bounded by the membranes, but in case of bacteria you can see that the nucleus is dispersed in whole cell. There is no true boundary between the cytoplasm, outer membrane and the nucleus. So this dispersion, this stuff basically denotes that their nucleus is not true. So here is the end of our lecture. Goodbye.